one thing that you'll come to realize is that the high frequency of the spectral power of heart rate variability is associated with the parasympathetic system, whereas the low frequency is associated with both the parasympathetic and the sympathetic nervous system. And I'm going to explain to you why that is. So in the SA node, which is, as we know, the pacemaker, there are 4 important ion channels on the SA node cell membranes. You have the sodium channel, potassium channel, actually 2 potassium channels, and a calcium channel. And within the cell, it is electrically negative relative to the outside of the cell. And so there's a 65 millivolt difference, so that the inside is 65 millivolts lower than the outside of the cell. So when you have positive ions move across the cell membranes through the ion channels, it neutralizes or decreases this electrical potential difference. And so what we say is that it depolarizes the cell membrane or the cell. On the other hand, if you have potassium channels, which goes in the opposite direction, it enhances this polarization, or it repolarizes or hyperpolarizes the cell membrane. And this is sort of a diagram to illustrate this. So you have a measurement of the voltage potential difference across the cell membrane. And as it goes up, from negative 65 millivolts to 0 and even positive potential difference, it's depolarization. And as it goes down, it's repolarization. The sodium channel has what's called a leaky current. And this leaky current causes a depolarization, a very slow depolarization of the cell membrane. And what happens is that slowly, as it reaches a threshold, as denoted by this dashed line here, it causes the calcium channel to open up. And this causes a rush of calcium channels into the cell, and then actively leads to the depolarization of the SA node cells. Subsequently, the potassium channels open up, and then it reverses this depolarization that is caused by both the sodium and calcium channels, and you get repolarization as denoted by this downslope of this potential difference. Now, taking this within the context of the autonomic nervous system, you could see how the autonomic system affects this pacemaking ability of the cell. So the parasympathetic nervous system involves the acetylcholine coming from the postganglion of the parasympathetic nervous system, binding to the M2 or the muscarinic receptor, activating the G protein, which then subsequently activates this potassium-associated acetylcholine channel. And because this is a potassium channel that goes in the direction that enhances the polarization, what it does is that during the stage of this slow leak of this sodium channel, it leads to this very slow rise of the depolarization. Now, this is in the dashed line is what is considered a normal heart rhythm. So if there was no intervention from either the sympathetic or the vagal system or the parasympathetic system, you would have this type of automaticity of the electrical potential. But with the vagal nerve, it slows this depolarization and subsequently leads to a slower heart rate. Essentially, you're not having firing of these cells happening more frequently. The sympathetic nervous system, on the other hand, operates through norepinephrine or epinephrine that binds to the beta-1 receptors. The beta-1 receptors then bind to adenylyl cyclase, which converts ATP to cyclic AMP. And the cyclic AMP hastens or enhances this leaky current in the sodium channel. And this leads to a rise of this depolarization, this steady leak, and then leads to a faster polarization or faster heart rate. The other thing that it does is that the cyclic AMP activates protein kinase A, then activates potassium channels and calcium channels. So what it does is it increases the depolarization, the fast depolarization and fast repolarization. So it enables a faster beating of the heart. So why am I harping on this, and why am I bothering you with all this details? Because it's really important to know this physiology to understand what you're seeing when you're obtaining the heart rhythms that you get from your various devices. The SA node is where both the parasympathetic nervous system and the sympathetic nervous system meet. And so we can gain insights in the activities of the autonomic nervous system based on the heart rate over time. And what we look at is the interbeat interval, or the RR interval. So this is an example of a EKG of a normal sinus rhythm. And we designate the RR interval. So R indicates the peak of this QRS segment. And then we identify the RR interval, the time between these intervals. And then we can obtain a calculation, a graph, which I had shown before, of the RR interval over time to get a better sense of what is happening with the parasympathetic and sympathetic nervous system. What happens, though, if you have okay? So what happens, however, when you have pre-atrial contractions, where you have contractions that's happening at not only the SA nodes, but also in the other parts, ectopic sites of the right atrium? You would see that the RR is designated here, but does the RR interval actually provide any information about the autonomic nervous system? And what happens if you have something called atrial fibrillation, where you have multiple ectopic sites throughout the right atrium and sometimes the left atrium that leads to this erratic heart rhythm? And what we recognize is that because a lot of these extra nodes occur outside the SA node, we consider them as noise and not reflective of what's happening in the autonomic nervous system. So the important thing to note is that heartbeats originating from the SA node are the only ones that provide sufficient insights into the parasympathetic nervous system and sympathetic nervous system. And to delineate what are sort of the normal rhythms through the SA nodes, we change R into Ns, or normal rhythms. And the way we know on an EKG whether it's a normal rhythm or not is we could see something called a P wave before this QRS. You can see these P waves. And these P waves are the electrical contractions or electrical signals going through the right atrium. And we know through the axis or the morphology, how it's shaped, that it's going through the right system. In the periatrial contractions, we notice that the normal sinus rhythms are occurring here because we see the P waves here. We don't necessarily see the P waves that occur before these preatrial contractions. And as a result, we eliminate these R waves. And sometimes what we do is we interpolate the N between the normal ones to get an idea of what the SA node would have done if it hadn't been for these ectopic beats. For atrial fibrillation, on the other hand, it's so inundated by all these signals that are coming from these other parts of the right atrium that we can't get enough information in the SA node. So we can't really get much information in patients with atrial fibrillations about their autonomic nervous system. So this is something that you can get from your EKG. However, in many of you, we're getting heart rhythms from use of watches. And the watches don't use, obtain EKGs. What we obtain are the PPGs, or the photoplethysmography, which measures the flow through microvasculatures in your skin. And it has, it doesn't have this rapid upstroke that you can see in the electropolarization. It's a slow rise. And the other thing to note is that you can't tell whether these things are normal sinus rhythms or whether they are attributed to an ectopic beat. I mean, you can't directly, because there isn't a P wave you don't have a P wave equivalent in the PPG signals. This actually is not a P wave. It is a reflective pulse wave, so it's not a P wave. So this is one of the challenges that you may encounter as you do your research, is that the PPGs won't give you information about or directly won't give you information about what kind of rhythm you have. And sometimes if someone has a lot of ectopic atrial beats or atrial fibrillation, it falsely gives you a higher heart rate variability. And it doesn't give you a reflection of your autonomic nervous system. The next thing the reason that this diagram is really important is to really give you an understanding of the dynamics of the SA node. In the SA nodes, the acetylcholine receptor operates very quickly. So the acetylcholine, after it binds to the M2 receptor, the G protein acts literally in few milliseconds to activate this potassium ion channel. On the other hand, the sympathetic nervous system, it's intubated through this enzyme called adenylyl cyclase. And this cyclase operates at much slower timescales, approximately 3 seconds or more. And as a result, it is much slower to respond. The other thing to note is that there are different dynamics in the neurotransmitters associated with each of these autonomic nervous system branches. The parasympathetic nervous system utilizes acetylcholine, and the sympathetic nervous system utilizes norepinephrine or epinephrine. And the acetylcholine, when it's released from this bulb of the preganglionic neuron, it is quickly eliminated by abundant presence of acetylcholinesterase within the gap junction. On the other hand, the sympathetic nervous system has to uptake these norepinephrine within the gap junctions. And the timescales are much different. So the parasympathetic nervous system removes these things about every 0.2 seconds, whereas the sympathetic nervous system removes it in 20 seconds. And what this leads to is a very different dynamic for each of these branches of the nervous system. For the parasympathetic nervous system, they've done this in dogs, is they've stimulated the vagal nerve. And you can see the stimulation right here. They essentially give 7 hertz electrical stimulation for about 22 seconds. And right away, you can see that the beats per minute or the heart rate significantly decreases. And almost immediately after you initiate the stimulation. And the moment it stops, you stop the stimulation, the heart rate jumps back up, nearly close to its original heart rate rhythm. And the same after you do the second stimulation. And so the parasympathetic has a rapid onset, quick execution, and a short recovery time. The sympathetic nervous system, on the other hand, when you're delivering 20 hertz stimulation to the sympathetic nerve and take a look at the heart rhythm, you can see that it doesn't kick in right away. There's a little bit of a lag, about 7 seconds or so, from the initiation of electrostimulation and then the rise of the heart rate. And this rise in the heart rate is not immediate like you see in the vagal system, but it's very slow. And after you stop the stimulation, there's also a lag, and it's a very slow recovery time. So the sympathetics has a delayed onset, slow execution, and a long recovery time. So what does this mean? Is that when we're breathing and are breathing at every 3 to 7 seconds or 9 to 24 cycles per minute, we're operating in the high frequency range. And as noted, parasympathetic nervous system is the only one that really is able to operate in the higher frequency range due to its dynamics. Quick onset, quick execution, rapid recovery time, whereas the sympathetic nervous system is simply too slow. So when you have something like respiratory sinus arrhythmia, the changes in your heart rate with respirations, it's predominantly a parasympathetic phenomenon. And it's simply due to the fact that the SA node has a filter, which inhibits the sympathetic nervous system to operate at a faster frequency.